In our last lecture, we finished up the central nervous system by talking about the brain, the spinal cord, and all that kind of good stuff. Now we're going to enter our peripheral nervous system. Now, as a reminder, here's where I suggested that you jump onto Blackboard and print out the PNS bonus slides. That might make your life a whole lot easier because some of these slides are not going to be in the packet. And another spot that you're going to want to print out slides, let me just find it here for a second, sorry. If you go into the Exam 3 folder, there are two documents that you may want to print. There's a special sentences document that you really must print, and there are PowerPoint slides for the special senses that you may want to. Those two kind of go together. So probably towards the end of this lecture or the beginning of next lecture, you're going to want to have those senses documents. But for the peripheral nervous system, you're going to want those PNS slides right now. Now, when we mentioned the peripheral nervous system at the beginning of the nervous system, we identified that it was made up of two types of nerves: cranial nerves, which there are 12 pair, excuse me, each are named and numbered, and you're experiencing that a bit with your lab content when you're looking at your olfactory, your, excuse me, optic, trigeminal, ocular, motor, those guys, those are all examples of cranial nerves that you're learning for the lab content, and they arise from the brain. They branch off of the brain. The other set of nerves are the spinal nerves. There are 31 pair of spinal nerves. Each will exit the spinal cord through the intervertebral foramen, that space, that gap between two vertebrae that are stacked one on top of the other. This gives us a total of 43 pair of nerves that make up the peripheral nervous system. Now, first thing we're going to do is compare the divisions of the motor or efferent division. So we're going to take a moment and we're going to look at what are the similarities, what are the differences between the autonomic nervous system and the somatic nervous system. Now, this is the first slide of two slides that are talking about a comparison between the somatic and the autonomic nervous system, okay? So if we look at our vertical columns, the one in green is labeled somatic. The one in I guess that's a purpley kind of color, is the autonomic nervous system. Then the first column is saying, okay, how are we going to compare these things? What are we going to look at? So we're going to look at effector organs, type of control, neural pathway, and those sorts of things, all right? So when we look at the effector organs, those organs that are going to be feeling an effect from either the somatic or the autonomic nervous system, we see that there's difference. We know that the somatic nervous system only deals with skeletal muscle, so that's going to be the only effector organ that will be innervated, serviced by the somatic nervous system. On the other hand, the autonomic nervous system, they've got quite a little grouping under there. We're going to find cardiac muscles, smooth muscles, and any and all glands that are going to be affected by neural control will be under the autonomic nervous system. Now, what type of control? Very simply, do you have conscious control over it or do you not? Your skeletal muscles, you have voluntary control. And we've said this before, not every time a muscle contracts do you consciously think, okay, I'm going to have that muscle contract. Most times they just kind of contract. But you have the ability to cause a contraction or prevent a contraction. Involuntary means, thank goodness, you have no control over this. Because if you think about it, if you had to have voluntary control over your cardiac muscle, that would mean you would have to remind your heart to contract every time it's going to beat. I would be dead within moments because I would forget. Okay, so what we mean by that is, do you have control or does your body just do it? And that's what we see for the involuntary control. Now, the neural pathway, don't let this get to you, all right? The bottom line is, when we look at our neural pathway, how many neurons service the autonomic nervous system or the somatic nervous system. When I'm looking at how many neurons go from the central nervous system to the effector organs, how many neurons are there? That's what we mean. So if we look under somatic, this is the one we're familiar with, right? This is the one we've been talking about in our last section. There's one motor neuron from the central nervous system. It synapses directly with the skeletal muscle. So the cell body is going to be contained within the gray matter of the spinal cord. The axon exits and forms the nerve, and that's where we're going to find, at the end of that one neuron, we're going to find our presynaptic terminals, or gecko toes, that are going to be in very, very close proximity to the skeletal muscle. It's one neuron, as compared to the autonomic nervous system. We'll break it down a little bit for you. Bottom line is, autonomic nervous system has a 2-neuron neural pathway. One motor neuron from the central nervous system synapses with a second motor neuron in a ganglion. Okay? In the ganglion, we're going to find the cell bodies of that second In that ganglion, we're going to find the cell body of the second neuron. That second neuron is what's going to synapse, connect with, have an effect on the visceral effector. Okay, so for voluntary, it's what you're familiar with the skeletal muscle. One neuron from the central nervous system to the muscle. For the autonomic nervous system, we have 2 neurons. The 2 neurons, the first and the second neuron, are going to synapse in a ganglion which is outside of the central nervous system. Okay? That second neuron is what's going to synapse or connect to the visceral effector, the effector organ, another way to say it, cardiac muscle, smooth muscle, and any or all of those glands that we've talked about. The action or the effect, and again, I kept the colors the same to kind of keep you thinking, all right, green is the somatic, purple is the autonomic. So what's the action on the effector? When an action potential, a nerve impulse comes along the somatic neural pathway, what effect does it have on the effector organ? And it's always excitatory. It always causes something to happen. However, in the autonomic nervous system, sometimes it's excitatory and sometimes it's inhibitory. Sometimes it causes something to happen. Sometimes it slows whatever's happening. All right, it may not cause it to stop completely, but it might slow it down. Think parasympathetic. Okay, the neurotransmitter for the somatic nervous system, it's always, always, always going to be our old friend acetylcholine. For the autonomic nervous system, it may be either acetylcholine or norepinephrine. Right, norepinephrine is going to be secreted by the adrenal glands. Right, it's another neurotransmitter. Next, we're going to look at and compare the 2 divisions of the sympathetic division. Goodness gracious. We're going to look at the 2 divisions of the autonomic nervous system, the sympathetic division and the parasympathetic division. You'll remember, hopefully, that when we talked about our sympathetic and parasympathetic, that they worked in opposition to one another. If the sympathetic division sped something up, this parasympathetic would slow it down, and vice versa. All right, so we have to keep that in mind, that they're kind of paired and working in opposition. Sympathetic division is fight or flight. Parasympathetic is the rest and digest. So again, this chart is set up the same way, right? We have sympathetic in red, we have parasympathetic in green, and in blue coming down the side is the characteristics we're going to compare. Now, before you freak out, in preganglion and postganglion, what is she talking about? Remember, we're talking about the divisions of the autonomic nervous system. We just discovered that the autonomic nervous system is a 2-neuron neural pathway. So you're going to have the ganglion in the middle. You're going to have a preganglionic fiber from the first neuron. You're going to have a postganglionic fiber from the second neuron. Okay, and you'll notice that in one instance, the preganglionic fiber is short, the postganglionic is long, and vice versa. Think of it this way. We're going to drive from Binghamton to Buffalo. Somewhere along the way, we're going to stop for lunch. Some folks are going to drive to Syracuse and stop for lunch. Then it's a short drive to lunch, and then they have a much longer drive to get to Buffalo. Other folks are saying, I don't want to eat that early. I want to get further. We're not going to stop for lunch until Batavia, which is much, much, much closer to Buffalo. So their drive before lunch is longer. Their drive after lunch is shorter. That's what we're talking about here. Okay, the preganglionic fiber is the drive from Binghamton to the lunch stop. The postganglionic fiber is the drive from the lunch stop to Buffalo. The distance is going to be the same. It's going to be, for the sake of argument, making this up, 500 miles. But how we chop it up is going to be different, and that's what we're talking about with our sympathetic and parasympathetic division. Now, in the sympathetic division, the preganglionic fiber is short, which means the ganglion is going to be, as we see here, the ganglion is going to be near the spinal cord because that preganglionic fiber is going to be short. It's only going to go a short distance. I'm only going to Syracuse. Okay? Now, if my preganglionic, my pre-lunch drive is short, that means my postganglionic fiber, my post-lunch drive is going to be long because I need to go the same distance. Sympathetic is going to be the opposite to that. This is where we're going to drive from Binghamton to Batavia before we have lunch. We're going to have a long preganglionic fiber, which means we're going to have a short postganglionic fiber. That means that our ganglion is going to be near or even in the effector in some instances. All right, so what the bottom line comes down to is we're going to have these two neurons, and we're going to have a ganglion. The difference is, where is the ganglion positioned? Is it close to the effector organ, or is it far from the effector organ? And that's going to determine the length of our pre- and post-ganglionic fibers. Now, NT is an abbreviation for neurotransmitter. Again, red is sympathetic, green is parasympathetic. The neurotransmitter that we're talking about for our sympathetic, all preganglionic neurons are going to use acetylcholine. A few postganglionic neurons are going to use acetylcholine. Most of the postganglion will use norepinephrine. As compared to, for the parasympathetic, all of the neurotransmitters in use will be acetylcholine. Now, what's the point? What do we use our sympathetic division for? What do we use our parasympathetic for? Sympathetic is the expenditure of energy, that fight or flight concept that we talked about. And the parasympathetic is to reverse that, to conserve and restore body energy, the rest and digest. Right, so really this is simply a comparison and overview of our parts of the autonomic nervous system. This brings us to our senses, because our senses will be considered in large part of our nervous system, obviously. When we talk about our senses, we have the ability to detect a sensation. Well, what do we mean by a sensation? The awareness of internal and external conditions of the body. Okay, what's going on in the body, and the ability to determine what those conditions are and if there has been a change in those conditions. When we look at sensations, we're going to be able to identify 4 characteristics, and they're going to be somewhat different depending upon which sensation we're talking about, which sense we're talking about. But the bottom line is all of our senses are going to be dependent on these sensations.